The first of these is from, three of these, is from Gemma in Oxford, hurrah, who's going to talk to us about normal cholangiocytes and cholangiocarcinoma in culture. Thank you.
Good morning, everyone, and I'd like to say a big thank you to the AMF for hosting this wonderful event. Today, I will be talking very briefly about my PhD research. So I'm currently a PhD student working with Dr. Shiji Kai and Professor David Kerr at the University of Oxford. And my research looks at the role of normal cholangiocytes in regulating cholangiocarcinoma cells. So to start off with, tumours do not grow in isolation. They're constantly interacting with the tumour microenvironment and the stromal compartment, exchanging molecular communications with localised fibroblast, endothelial cells, and immune cells. A compartment often overlooked is the contribution of the neighbouring non-cancerous normal cells. These cells, often directly adjacent to the tumour, play a unique role in preventative or permissive state of the tissue. We hypothesise that surrounding non-cancerous cholangiocyte cells interact with cholangiocarcinoma cells during the very early onset of tumour development, and that this interaction causes key alterations to gene expression, metabolism, and ultimately the behaviour of cholangiocarcinoma cells. Therefore, we decided to investigate the effect of paracrine signalling on cholangiocyte cells and cholangiocarcinoma. We used a 2D Transwell system to evaluate the behaviour change of our cholangiocyte cells when grown in indirect co-culture with cholangiocarcinoma. We used an intrahepatic cholangiocarcinoma cell line CCSW1 and an immortalised cholangiocyte cell line MMNK1. Particularly interesting, we used a Transwell system with a 0.4 micron pore semi-permeable membrane. This means that there are tiny holes in the system that we grow between our two cells. These cells are 0.4 microns wide, which is far too small for the cells to move between, but large enough for small secreting molecules to move between. Therefore, we can kind of interpret the almost long-range communication between these two cell lines. So to kick off then, let's look at the cholangiocyte compartment. Using our co-culture system, we found that at 48 hours, co-culture actually promoted cholangiocyte viability. And we saw a significant increase in MMNK1 cell viability. We also saw an increase in the proportion of cells in the G2M phase of the cell cycle, and a significant decrease in the proportion of apoptotic cells, shown here. We then started to evaluate the molecular mechanism behind these behavioural changes, and we saw a significant decrease in the protein expression of the apoptotic markers such as P53, BAX, caspase-3, and caspase-8, shown here. Furthermore, we then looked at how we could evaluate the gene expression changes in our cholangiocyte compartment when grown in co-culture. To do this, we used bulk RNA sequencing to identify changes and have used evaluation of two databases, the Hallmark database and the KEGG database, as pathway database choice can influence actually gene signature detection. So we used both to validate our results and get a more biologically plausible readout. And what we found was that when we looked at the pathways significantly enriched in our MMNK1 cells, we saw upregulation in gene signatures for the G2M checkpoint, and also downregulation in gene signatures for the apoptosis and P53 pathways, which was in agreement with our previous work. We then looked at the same RNA sequencing using the KEGG pathway analysis. And interestingly, we found an upregulation in gene signatures for drug metabolism, cytokine P450, which will come slightly in later, as well as a decrease in P53 signaling and a decrease in cytokine-cytokine receptor interactions. What was really interesting here is that we saw a significant decrease in gene expression profiles for glycine serine 3' metabolism and alanine aspartate glutamate metabolism. And due to this, we decided to investigate whether this was replicated within the cholangiocyte secretome. So we then conducted a large-scale metabolomic analysis, which is where we looked at the intracellular metabolites in our cholangiocyte cells at 6, 24, and 48 hours. I've only shown 48 hours here for time. So on the left, you can see the pathway analysis that we conducted using these metabolites at 48 hours. And two of the pathways that were significantly enriched was alanine, aspartate, and glutamate metabolism, and glycine, serine, threonine metabolism, which again is in agreement with our RNA sequencing data. From here, we then looked at the individual metabolites themselves. This volcano plot in the middle demonstrates the metabolites in MMK1 cells at 48 hours that are significantly upregulated in red or downregulated shown in blue. So we can see that creatine and choline were significantly increased in concentration, whilst glutamine and aspartate were significantly decreased in concentration. We then took these metabolites forward to investigate whether we could replicate or mimic the effect of co-culture using these exogenous doses of metabolites. And what I'm showing here is the effect of creatine. So creatine is actually used in cells as a store of ATP, ATP being the energy currency of the cells. And what we found is when we went back and looked at our co-culture system, when our MMNK1, so our cholangiocyte cells, are in co-culture with our intrahepatic cholangiocarcinoma cell lines or our extrahepatic cholangiocarcinoma cell line, we saw a significant increase in the ATP concentration of the cell. When we then looked at exogenous doses of creatine, we could mimic this co-culture increased concentration of ATP. So when we added exogenous creatine, we actually could increase the ATP concentration within our cholangiocytes. And likewise, exogenous creatine actually improved the cell viability of our cholangiocyte cells. And therefore, we hypothesized that during this co-culture, it is creatine that is responsible for the increase in ATP that we see, as well as the improved in cell viability. But this isn't the whole story. So now I'll briefly go through our cholangiocarcinoma compartment, which was using the intrahepatic cell line CCSW1. So similar to the cholangiocyte cell line, we looked at the functional changes that we saw. And interestingly, we saw a significant decrease in cholangiocarcinoma cell viability, which is not what we expected. You know, we expected to see the cholangiocarcinomas outperforming our cholangiocyte cells. But at 48 hours, which is our time point, this is not what we saw at all. So we saw a decrease in cell viability. We also saw a significant decrease in the proportion of cells in the S phase of the cell cycle. And we saw a decrease in the proportion of migratory cells. So here you can just see some staining images of cells grown in monoculture and then grown in cold culture. We saw a reduction in the proportion of migration. So we then looked to try and explain what were the molecular reasons for this. And we measured the expression of EMT markers at a protein and a gene level. And we found that actually at both the protein and gene level, there was a reduction in the EMT markers such as bimentin, slug, and also snail. So we can see that from the blot here, just quantified here. This occurred concurrently with an increase in both the gene expression and the protein expression of ecadherin. So we hypothesized that this molecular change is what's causing our reduction in migration of our cholangiocarcinoma cells we saw earlier. So as I mentioned, for our cholangiocyte cells, we did this bulk RNA sequencing and used two different databases to analyze our result. Interestingly, what we saw in the CCSW1 cells was out of the 11 significantly enriched gene pathways, 4 of these represented cytokine signaling. So we had interferon alpha, interferon gamma, IL-6, and tumor necrotic factor alpha signaling. When we looked at the same gene expression data using the KEGG database, again, we saw this significant increase in cytokine-cytokine receptor interactions, as well as an increase in chemokine signaling. Therefore, we decided to investigate whether this significant change to cytokine gene expression was mimicked in the cholangiocarcinoma secretome. So that's what we did. We used a human cytokine array to evaluate the expression of 42 cytokines in the secretome of our cholangiocarcinoma cells when grown alone and when grown in co-culture. And what we found was there was a marked difference in what concentration of these cytokines the cholangiocarcinoma cells were exposed to before and after co-culture. So we can see here, one of the most striking was IL-6. So when our CCSW1 cells were grown alone. They had a very low exposure to IL-6, but when they were grown in this co-culture conditions, there was a massive increase in the IL-6 that they were exposed to. And this led us to think that perhaps, as typically thought, the IL-6 concentration that leads to chronic CCA development is not only provided by the fibroblast and the stromal compartment, but actually provided by, in part, the cholangiocyte cells that can surround the tumour. Moving forwards, in the recent publications and the previous work that's been conducted, it's been noted that IL-6 can actually be modulated epigenetically. And in terms of chronic IL-6 signaling can occur from epigenetic changes to STAT3 and especially suppressor of cytokine signaling 3. Therefore, we decided to look at a panel of epigenetic inhibitors. Could we then modulate our cholangiocarcinoma behavior using epigenetic modulation? On the left, this shows a heat map of the panel we conducted. So the panel, we had 63 compounds obtained from the Structural Genomics Consortium, and we used our 4 CCA cell lines as well as our cholangiocyte cell line. We can see that yellow or white is better cell viability, so up 100%, and black and purple is poor cell viability. And what we wanted to select was a compound that caused a large decrease in CCA cell viability, but actually that our MMNK1 cells, our cholangiocyte cells, were mostly resistant to. So from here, we selected these 2 compounds, UNC642 and STC3027. So UNC642, a methyltransferase, and STC3027, arginine transferase. So as we've heard from today, these are just some IC50 curves, so we look at how good the drug is at killing our cells. And we first did that in our cholangiocarcinoma cell line, so how much could our drugs kill our cancer cells? And for UNC642, it was about 5 micromolar, and for STC3027, it was about 6 micromolar. What was really interesting is then we decided to use these drugs in our co-culture system. So our cholangiocarcinoma cells grown indirectly with our cholangiocyte cells. And what we found was that actually this dramatically increased the IC50 for these drugs. So the concentration of these drugs needed to kill the cholangiocarcinoma increased, suggesting to us that actually these cholangiocyte cells can in part contribute towards the epigenetic inhibitor resistance of cholangiocarcinoma cells. So that was a bit of a whistle-stop tour, but in summary, my research, along with Dr. Shiji Kai and Professor David Kerr, has looked at how interactions with the non-cancerous cholangiocytes can actually modulate the cholangiocarcinoma cell survival, gene regulation, and metabolism at the very early onset. And that this alterations via cholangiocytes can actually have serious implications for downstream targeting and also the development of novel chemotherapeutics. I would like to thank my supervisors, Dr. Shiji Kai and Dr. David Kerr, along with all of my fantastic collaborators, as well as the AMF for funding this project and for everything they've done for us. Thank you.